So here we go. It's CVPP day. It's Tuesday. It's week 2. It's the winter of 2022. Here we go. Where have we been? What have we been talking about? Endothelial cells mainly. Remember, those are the cells that line the blood vessel are in contact with the blood. They are everywhere. All the blood vessels. The heart has them. Capillaries are made of them, so they're everywhere. We also started talking about these endothelial cells secrete all kinds of stuff, all kinds of molecules and substances. Both some of these substances diffuse into the tunica, the tunics, into the vessel wall and have effects. Some of them diffuse out into the bloodstream. Some of them kind of stick on the outside of the endothelial cell and are in contact with the blood. But the ones we're concerned about are the ones that help prevent thrombosis. And we said when your blood clots, that is officially called thrombosis. We talked about the slippery 3, which you should definitely know: thrombomodulin, prostacyclin, and tissue plasminogen activator. And we also said that the endothelial cells are important if you're bleeding. They secrete things like von Willebrand factor, which is very important. We'll talk more about that in a second, but very important in helping your blood clot. And endothelin I could have added in there as well. Then we said endothelial cells, if they get injured and irritated, usually from hypertension, or maybe you went to the dentist and you got a little bacteria, some bacteria have gotten into your bloodstream and stuck to the endothelial wall of, let's say, your popliteal artery and caused an inflammation that's called a vasculitis. Those endothelial cells are upset and injured as well. So there's other things that can injure cells. But if they get injured, strangely, they do two things. They undersecrete the slippery 3, so that increases the chances of getting a blood clot. They also oversecrete von Willebrand factor, and that's a powerful blood clotting agent. It's a member of the blood clotting cascade. And that's kind of where we left off, and we need to talk more about this von Willebrand factor. I guess we should know who makes it first of all. There's a cute little vesicle that floats around in the endothelial cells, and these are called Weibel-Palade bodies. Weibel-Palade bodies. Not like these Weibels of day and age, but I mean it's pronounced the same as Weibels. And they're found everywhere in all endothelial cells. And those are what actually make von Willebrand factor. So note card, what makes von Willebrand factor? Weibel-Palade bodies of endothelial cells. All right, so who cares about a Weibel-Palade? Well, they synthesize, they store, and they secrete von Willebrand factor. Von Willebrand factor gets out into the bloodstream and promotes something called hemostasis, or blood clotting, or the process of thrombosis. So that's, of course, hemostasis is incredibly important at stopping hemorrhage. So because it's a blood clotting agent, it's considered a prothrombogenic molecule, or you could say a thrombogenic molecule. Prothrombogenic is better because pro means yes, it does this. Thrombo is blood clot. Genic is the process of. So promotes the process of thrombosis. Okay, so what does von Willebrand factor do? There's two things. There's really more than that, but it acts as a flypaper. It sticks on the outside of endothelial cells in the flow of blood and catches platelets, and blood platelets start sticking to the area. Von Willebrand factor is secreted classically in a rip in the blood vessel. Let's say you ripped your blood vessel from doing something and it's leaking blood out into the interstitium. Von Willebrand factor is secreted, and it sticks to the ripped area and starts catching platelets to plug the leak. Kind of simple, actually. It also acts as a scaffolding, and platelets can build up and grow on it, and this blood clot or this thrombus can grow on the base, and at the base of this whole thing is von Willebrand factor. It also does another important thing. It chaperones and protects a coagulation factor called factor VIII. Factor VIII is also very, very important in the blood clotting cascade. So without von Willebrand factor, factor VIII doesn't work. So you have a tendency to bleed if something's wrong with von Willebrand factor. Okay, so note, factor VIII will not be very effective in the coagulation cascade without von Willebrand factor. All right, so that brings us to von Willebrand disease. So what the heck is that? It's a genetic disease. It's caused by a mutation on chromosome 12. We won't go any deeper than that. But it's considered a bleeding disease. And it's a mutated von Willebrand factor. So without von Willebrand factor, and you get a cut and start bleeding, you're going to have trouble stopping the bleeding. Okay? So the mutation causes a transcription of a mutated von Willebrand factor. So the central dogma is messed up. So von Willebrand factor in and of itself doesn't work, so you're going to have trouble clotting. But remember we said von Willebrand factor chaperones factor VIII. So factor VIII, even though there's nothing wrong with it, it doesn't work well because von Willebrand factor is not there to shelter it and help it work better. So that's kind of a double whammy type thing. Worsens the blood clotting problem. Those affected with von Willebrand disease, they're said to have dysfunctional hemostasis. So they can't clot the blood. So let's do some fun facts here about von Willebrand disease. It is the most common inherited or genetic related bleeding disorder, period. It's rare, 1% of the population, but that's not crazy rare. But most people have it and they don't even know that they have it because it's not severe. You can have different stages of, or different degrees of mutations. Some of them are not that big of a deal. You might bleed a little bit longer than normal, but you don't even notice it. But about 0.01% of people with von Willebrand disease are severely affected by it. And these are the ones who have significant morbidity. Their lives are not great. Von Willebrand disease is also much more common than the hemophilias. But I ask this question on every test, so here's a test question for those of you who are listening. Von Willebrand disease is not a hemophilia. It is not. It's a different animal. It's a mutation of von Willebrand factor. It's not considered a hemophilia. Right, more fun facts. There are 4 subtypes of von Willebrand disease. Some are really bad, some are not so bad. We're not going to go into the weeds any farther than that. I think that's too deep. But the patients, that 0.01% of patients can become severely ill and frequently bleed and need numerous transfusions. And they're constantly going into the emergency room. And when they get there, how do you treat people with von Willebrand disease? Well, you give them von Willebrand factor. That should be VWF. Plus, you have to give them factor VIII as well. And there's other things, but that's kind of the gist of it. Now, you don't have to know this whole table, but I think you should know one hemophilia. The most common type of hemophilia, which is another bleeding problem, actually occurs because of a deficiency of factor VIII. We said factor VIII was a very important player in the blood coagulation cascade, and without it you have a lot of trouble clotting blood. And so deficiency means it's not being produced enough. It's not mutated, it's just the endothelial cells just don't make it very good because of mutations and problems within, but it just doesn't, you're deficient in factor VIII. Represents about 83% of all hemophiliac 83% of all hemophiliacs have this problem, hemophilia type A. Hemophilia type B, we don't need to worry about. Well, should you worry about that? I think that you probably should know that too. That's a deficiency in coagulation factor 9. So 8 and 9 are the most common. And that's all we'll say about that. And then again, von Willebrand disease, we already talked about all this. Type O blood may increase the risk and severity if you do have von Willebrand disease for reasons we won't get into. Okay, what about hemophilia versus von Willebrand disease? This is always a good slide to know. So von Willebrand disease, that should be VWD, typically affects mucous membranes in the skin. Hemophilias, typically the bleeding occurs inside of muscles or joints, or even inside internal organs like the liver and the kidney, things like that. But von Willebrand factor, mucous membranes are everywhere, right? In the nasal cavity, the oral cavity, the mouth, the nose. The entire intestinal tract is lined with, it's considered a mucous membrane, endothelium, or the simple columnar cells. And so the vagina, the uterus are all lined with a mucous membrane. And so you get bleeding from all these structures, have potential to bleed. What are the classic symptoms of von Willebrand disease? Nosebleeds is one sign. And everybody, especially kids in the winter, they get nosebleeds. Not that big of a deal. But these people are getting bleeds every day or a couple times a week. So that's unusual. Periods that go on and on and on, really prolonged periods could be a sign. Hematochezia or melena, and we'll talk about those in GIGU, but hematochezia is red blood coming out of the anus, usually with bowel movement. Melena is a very dark, tarry looking stool. The blood has been degraded. So it depends where the bleed is occurring in the GI tract. We'll cover that more when we get to GIGU. And so it's not uncommon for people affected with von Willebrand disease to become anemic and have anemia from this condition. So SARS-CoV-2 and COVID-19, there's a relationship here to von Willebrand disease. So people severely ill and people who have died with COVID-19, research has shown that they have a greatly increased level of von Willebrand factor in the blood. So they're prothrombogenic. In fact, over 500% increase has been shown. So, and a 300% increase in factor VIII has also been found in these patients. And that's what's killing them. It's really blood clots, micro blood clots that are killing them. So widespread thrombosis throughout the brain and the cardiovascular system. It's mainly in the pulmonary and the lungs, but it can be everywhere. But it's thrombosis that is killing patients. Right? So let's dive into that a little bit more. What is causing the increased von Willebrand factor and creating this prothrombogenic state? What is doing this? So the short answer, it's more complicated than this, but the short answer is endothelial cells are producing a nasty, nasty pro-inflammatory, super pro-inflammatory molecule and a super blood clotting molecule called superoxide. And that's a free radical. That's a radical oxygen species. And so why does this increase so much in people who get SARS-CoV-2? Some of you may have figured this out already. So recall that the angiotensin 1-7 is the good guy and angiotensin 2 is the bad guy. There's two branches of the R2A system, right? There's the angiotensin 2 part, and then there's the counteractive angiotensin 1-7 that has the opposite effects of angiotensin II. And they both bind to receptors all over the body. They're right next door. Angiotensin 1,7 uses the MAS receptor. Angiotensin II uses confusingly binds to the angiotensin 1 receptor. Okay, I don't know, that might be too deep. But yeah, so the receptors are there, and they have opposite effects of each other. Recall that angiotensin 1,7 is created by an enzyme called ACE2, not ACE. ACE is the one that you probably know well. That's in the lung microcirculation, and that creates angiotensin 2. But there's another enzyme called ACE2, and what that does is it grabs excess angiotensin 2 out of the blood, and it converts it into Marshall Dillon here, the good guy, angiotensin 1,7. Right? So recall excess angiotensin 2 is grabbed by ACE2 and converted into the good guy. And that way we don't have too much of that angiotensin 2 raising hell throughout the body. Okay, so but here's the problem. SARS-CoV-2 binds to ACE2 and wrecks it or clogs it up. So if ACE2 is busy fighting off SARS-CoV-2 virus, how's it going to take angiotensin 2 out of the bloodstream and convert to angiotensin 1,7? It's not. So you get a buildup of angiotensin 2, which is prothrombogenic, and you get a deficiency of angiotensin 1,7, which is antithrombogenic. It's an anti-blood clotter. It's an anti-inflammatory. And so that is the problem. It creates a prothrombogenic state that way, and it does something even more. So we also know from our studies that angiotensin 1,7, it is what prevents the formation of superoxide. Right? You remember free radicals? I think Dr. Doe talked about them. I'll show you the chart here in a second. But without angiotensin 1,7 around, you're going to get an overproduction of superoxide. And superoxide is a powerful inflammatory, so you'd get an inflammation in the lungs where SARS-CoV-2 is. Right? Because there's no angiotensin 1,7 in the lungs now. So you get an inflammation, and it's prothrombogenic, and it causes the overproduction of von Willebrand factor. Right? So that's kind of a double whammy with regard to thrombosis. Right? So increased superoxide encourages thrombosis by stimulating the overproduction of von Willebrand factor. To make things worse, superoxide decreases nitric oxide production, which stimulates arterioles and venules to vasoconstrict, so you have very slow blood flow through the capillaries. And that encourages you, anytime you get slow blood flow, that makes it easier for that blood flow to clot. And plus, if you get slow blood flow through the microcirculation, that's going to decrease gas exchange in the lungs. You're not going to be able to dump off carbon dioxide and pick up oxygen very well. And that leads to a progressive destruction or a knocking out of action of the alveoli. And you lose more and more and more alveoli. Pretty soon you can't breathe and you basically suffer or suffocate. And that's called acute respiratory distress syndrome. Your body becomes hypoxic and you die. Okay. Let's see. Yeah, we just said that. And the slow-moving blood through the capillaries encourages thrombosis because it's easy to clot blood that's moving slow. Right? So SARS-CoV-2 knocks out the ACE2 receptor. Without ACE2 receptor, you have a buildup of angiotensin 2. You have a decrease of angiotensin 1,7. You have an increase in the free radical. and you get endothelial cell dysfunction, increased von Willebrand factor, thrombosis, and then ARDS. It's kind of a flow chart there. So, and you don't have to memorize this whole thing. You probably did for biochemistry. At least some of you did. But here's the problem. We have oxygen. We have an enzyme called NADPH oxidase, which oxidizes oxygen, adds an electron to it, and you get this nasty superoxide molecule, which is a free radical. And normally, angiotensin 1,7 binds to NADPH oxidase and knocks it out of action or slows it down, so you don't make superoxide, right? And then we could go through this whole thing. You don't need to. For me, you don't need to worry about that, but you should know that angiotensin 1,7, which is made from ACE2, or made from the ACE2 enzyme, and you should know this part of the pathway. Angiotensin 2 is normally converted into angiotensin 1,7 by ACE2, and angiotensin 1,7 blocks the function of NADPH oxidase, and therefore you don't get much superoxide around. But SARS-CoV-2 blocks ACE2. If you block ACE2, this isn't around, and this process runs this way, and you get superoxide and hydroxy radical as well. All right, should be kind of review stuff for you. All right, so let's get into some easier sledding here. We still need to talk about, we talked all about the tunica intima. Let's talk about quickly the tunica media and the tunica adventitia, just to finish our little histology review. So tunica media, of course, is the middle of the artery or vein. It's the smooth muscle. It's where recoil, we've already talked about how when systole occurs, an artery is stretched out, and then during diastole it contracts. And so tunica media, in your brains you probably think tunica media is smooth muscle, but there's a structure to this, and it's not only smooth muscle. There's collagen, there is elastin, a stretchy material, and then there's these elastic laminae, these sheets of Swiss cheese. Okay, and here's the Swiss cheese. We talked about these Swiss cheese layers. Remember, this is the internal and external laminae. That's how you can tell this is an artery, right? Because veins don't have this. It's for protection, they think, because this artery is under higher pressure. Here's the endothelial layer we've been talking about. Now we're talking about this pink layer here, the tunica media. So let's look exactly at how this is really built. I mean, it really doesn't look like just smooth muscle cells. So you can see the smooth muscle cells where the S is. So this is the full thing. This is kind of naked here. They took stuff out. This is what all of these layers look like. But it kind of looks like a mess here. You can see the smooth muscle. But if we kind of break it down a little bit, you have inside your tunica media, there's these sheets of Swiss cheese called elastic laminae. And the sheets are connected together by different things. So we have these elastic fibers that connect these sheets together. So if you stretch this out or compress this down, these will snap it or push it back into shape. They like to be in, this is their native conformation here. So if you expand this and smash this out and stretch it, they'll snap back into its normal shape. Then we can add some collagen in here. There's collagen fibers connecting these layers of Swiss cheese together so they don't overstretch or overcompress. This is when aging has deleterious effects on this collagen. It makes it stiff, so you're not so springy anymore. And then we can add in the smooth muscle as well and put it all together, and it looks like this. So we have these elastic laminae, which are connected by these elastic fibers, which are also connected by strands of collagen, and then smooth muscle. So that's basically the wall. And therefore, it's very springy and stretchy. It likes to return to this shape. If you smash this thing flat, it'll spring back into this native shape when the pressure is relieved. So there is some recoil to this. And we've already said that recoil is very important during diastole because the recoil pushes blood downstream and keeps your blood constantly flowing, and it pushes blood backwards to close the semilunar valves. Right? We've talked about that. When systole occurs, let me try this little drawer here while I'm got it out. So when systole occurs, let's use the venous side for right now. This stretches way out like this. And then when diastole occurs, because of the design of the tunica media, it snaps back into its normal shape. See how that works? And then what happens to the blood? This section of blood. Some of the blood is shot this way and goes through this would go into the lungs, but same thing happens with the aorta here. Goes to the body. But some of the blood is shot backwards toward the heart. And luckily we have the semilunar valves, which are like little parachutes, and they pop open and the blood is caught by these things. And yeah, it shuts your semilunar valves that way. We've talked about that. Let's see, I think I have to do that. All right. So what about aging and hypertension? Now that we know about this tunica media, what's the relationship between them? So as people age, they get more layers of Swiss cheese in the tunica media. So they actually grow more layers. So that does a couple of things. It makes the artery more stiff and less springy. And because the tunica media increases in size, the lumen of the blood vessel decreases in size. Right? So that causes hypertension. Another thing happens is those collagen filaments, they tend to swell up and get thicker and stiffer. And so that also makes the artery less springy and has less ability to recoil. It's less elastic. Okay, so those are two mechanisms. You should know these two mechanisms of why hypertension occurs. Okay. Yep, so everything I said, a stiff and thickened non-elastic aorta will cause problems. Also, what's going to close the semilunar valves? I didn't mention that. So if it doesn't snap back, the semilunar valves don't close very well, and you get more leakage of blood or regurgitation of blood into the ventricles as you age. And we said the hypertension we've talked about. All right, let's talk about the very outer layer of the blood vessel. So we talked about the tunica intima, tunica media, and here's the tunica adventitia. So that's the outer layer of a blood vessel. It's made by connective tissue, mostly collagen fiber. It's got fibroblasts in it that create all these cells. And it's separated from the tunica media by the external elastic laminae. That's an easy histology question. Remember, that's not in veins, though. It's only in arteries. There is a covering around the outer surface, a connective tissue covering, which is important. Sometimes if you rip through the wall of the vessel and you start leaking blood, and this can save you. It can catch the blood. It's pretty tough. It's hard to get through, so it's like an extra protective layer there. These cells that live in the tunica adventitia are mainly fibroblasts, and we know fibroblasts make collagen mainly. They make elastin too. They make other stuff. Those are the two big ones. There's a little elasticity in the tunica adventitia as well. It's relatively thin. It's not as thick as the tunica media. The important thing of it here, let's go right to this picture. In large vessels like the aorta, this is living tissue, right? These are fibroblasts. How do these fibroblasts get serviced? How do they remove How do you remove waste? How do you get the little fibroblasts, which you can't see? We could draw little dots in there, but how do they get food? Well, they can't. Here's oxygen, right? This is an artery. Oxygen and nutrients. They can't go through the endothelial cells and soak all the way to the tunica adventitia. They can go about halfway to the tunica media, these nutrients. But so we need pipes. We need a circulatory system to service the outer part of blood vessels. And that's where the vasovasorum come in. So there's an artery and there's a nerve that live within arteries, big arteries. So it's like a vessels within vessels, and that services the outer half of the wall of a blood vessel. There's also a nervy vasorum, which is mainly sympathetic fiber. We've talked about that, and we said norepinephrine is released from these little nerves. That soaks in and causes muscle contraction. Right? We've talked about that. So I think I got all of this stuff. We'll look at a dissecting aneurysm more, but that's important. So yeah, it's blood vessels within blood vessels. That's called the vasovasorum. Arterial vasovasorum, venule, or a vein vasovasorum. You could be a little more specific with those. All right, there's different types of vasovasorum that you might have. Everybody's a little bit different. This one comes from the outside. It doesn't come from the inside. So that's called a vasovasorum externa. There's a different flavor that some people have called a vasovasorum interna, which doesn't come from the outside at all. Sometimes you can have blood vessels, a little new circulatory system growing from the tunica intima inward, all the way to the tunica adventitia. So that's another system that's called a vasovasorum interna, and then some people have a mix. Here's an example of a mixed vasovasorum. This is an arterial vasovasorum, but you can see that we have a blood vessel coming from the outside servicing the vessel, and we also have one that's grown from the inside, and so that's a mixed type. So that vessel is very happy, very well nourished. But here's the different types of them. Here's an externa type where the blood vessel comes from the outside. Here's an interna where it only comes from the interna, and we showed you a mixed one. And here's an externa. This just happens to be a venous vasovasorum. All right. So why am I bothering you with this pathology or histology stuff? Because there is pathology associated with these. What if you, let's do some drawing again. What if you, we'll make the blood blue so we can see it. What if you get an aneurysm? Well, let's say you get an aneurysm right here. I know we haven't talked about those, but aneurysm is a big outpouching of the blood vessel. Aneurysms can rupture. And what happens if it ruptures and this blue blood starts to build up here? Well, the pressure will start to rip through the tunica media. So we got this blood ripping and ripping and ripping down, and it can also eventually rip right into the main lumen, and that's called a dissecting aneurysm. So it's a certain type of dissecting aneurysm, which we will talk about more in the future. But vertebral artery and carotid artery dissections tend to be of this type. They tend to occur from vasovasorum. Right, so a little look into the future. Everything I said right there. And here's just a picture of it. Right. So yeah, that can cause all time we'll talk more about that in the future. All right, and we said already the nervy vasorum. That's how I always learned it. You can also call it the nervy vascularis. That's the yellow thing here. So these are autonomic nerves, usually sympathetic, specifically postganglionic sympathetics. And we said that norepinephrine is released from these things when the blood pressure drops in the carotid sinus. We said that you get an increased sympathetic outflow from the vasomotor center, and the sympathetics fly into these little terminals. They release norepinephrine. Norepinephrine soaks into the tunica media and stimulates it to contract. That narrows the lumen of the vessel, and that increases blood pressure. Right, everything I said. Norepinephrine's released from these things. Yep. Microcirculation, we've kind of talked about that. It's arteries, arterioles, venules, and the capillaries in between. There's also part of the microcirculation are these metarterioles, which we should mention. They are everywhere in the body. So let's take a look at these metarterioles. So here's how most capillaries, according to Guyton anyway, here's how most capillaries in the body are. The capillaries don't come off arterioles. Usually they come off a central tube that connects the arteriole to the venule, and the capillaries arise from this central tube, and this design is called a metarteriole, right? And this central tube is called the thoroughfare channel of the metarteriole. And we have on the arterial side, we have all these little smooth muscle sphincters here. So if this tissue, let's say this is your big toe, if this tissue does not need, or well, let's say the big toe tissue hasn't been fed in a while and it's getting hungry, it needs some oxygen, it's got some waste that needs to be moved out, it needs some glucose. If that's the case, there's messages that are sent, and we won't get into the details of that, but it sends signals to these sphincters to open. And so blood flows into the big toe tissue through these capillaries. And we've talked about high hydrostatic pressure, how it pushes out glucose and nitrogen and all that stuff. Blood fluid is pushed off here. And then down here, the hydrostatic pressure is much less, but oncotic pressure rules down here. So waste is sucked in back down here. And the tissue is serviced. Once the tissue is serviced, these capillaries shut, and that way we're not wasting blood into tissue that's already been serviced. A key point to understand, right now in your body, most of your capillaries are shut. You can't have all your capillaries on in your body at the same time. You would die of a hypovolemic shock. Too much blood would go to your skin, your brain, your muscles. You would die. There'd not be enough blood for flow. So it's an important concept to know that these capillaries are regulated. You can't have them on all the time. And we've already talked about someone who's bleeding to death, how epinephrine, amongst other things, goes to these and shuts these things down, right? Epinephrine can shut off the blood to the skin, if this is skin of the big toe we were talking about. Yeah, you can shut it down so the blood is conserved for the core and the other organs, the brain and the liver and the heart and such. We don't need to turn on the skin or the muscles for that matter if you're bleeding to death. So epinephrine can also shut these little capillaries down. All right, let's start the concept of body fluid distribution now, because we're heading towards swelling. But let's review our body fluids. So body fluids are present in the cells, the spaces outside of cells, and the spaces inside of blood vessels. And of course, blood fluid and interstitial fluid is very important for servicing tissue all over the body. Right? It allows for the transport of glucose, nitrogen, all those metabolites, and it also moves waste products. Lactic acid comes to mind. Lactic acid moves out, so it services tissue, as we've already said. There are two main compartments, or fluid compartments, in the body. There's the intracellular, so intra means inside the cell, and the extracellular means outside of the cell. So those fluids found either inside all your cells or outside all your cells. Of course, this extracellular fluid, we can break that down a little more specific. But intracellular fluid, that's all the fluid inside the cells. And there's billions of cells in the body. So most of the fluid, about 66% of your body fluid or your body water, actually lives within the intracellular fluid compartment. It lives within cells. The rest lives in the extracellular compartment. 33% of body fluid lives in this extracellular fluid compartment. Now there's three subcomponents of this extracellular fluid compartment. There's the blood plasma compartment, the interstitial fluid, which we've already talked about, and then there's this transcellular compartment, which we need to talk about. So the blood plasma compartment is easy. That's the space, that's the blood. It's the blood or the lymph fluid. It's the space inside blood vessels, inside the lymph vessels. And it contains blood and lymph fluid. Blood is sometimes better thought of as blood fluid or blood water because it's mostly water. It does not include fluid inside the blood cells, though. Like if there's fluid inside a red blood cell and white blood cell, that's still considered intercellular fluid. All right, so blood fluid gets pushed around. Once it gets pushed into the interstitium, though, we have a different compartment. We have the interstitial compartment. And the blood fluid changes names to interstitial fluid. It's still the same stuff. And then the interstitial fluid some of it gets sucked up by the lymph capillaries. It becomes lymph fluid. Still the same stuff, just different names. Interstitial fluid compartment we talked about. It's the space between cells and capillaries mainly. It's the largest of all the extracellular fluid compartments, and it contains interstitial fluid. And we've said this before, but just to reiterate, the blood or the interstitial fluid is held by proteoglycan molecules, so it's a gel. 99% of blood fluid that becomes interstitial fluid is held in a proteoglycan gel, very similar to a young nucleus pulposus. That way it discourages water to be wasted and squished around. It also acts as a reservoir. If you are hypernatremic and you need more water in your blood, you can tap into that reservoir, that proteoglycan gel, and you can remove fluid out of there to kind of get the balance between salt and water back to normal. Transcellular compartment is clinically important. So this is only 1% of extracellular fluid lives in this transcellular compartment. And you're going to know some of these structures. Transcellular fluid lives in the abdomen. We call it peritoneal or peritoneal fluid. It lives in the pleural space, pleural cavity, call it pleural fluid. It lives in the pericardial cavity. We call it pericardial fluid there. It's all the same stuff, though. But these are all examples of the transcellular compartment. It also, your ankle joint, your knee joint, your wrist joint, your elbow joint, the synovial fluid in there, that's a compartment. That's the transcellular compartment as well. Sweat, urine, renal tubular fluid, GI fluid, aqueous humor, cerebrospinal fluid. These are all members of the transcellular compartment. Right, just a cartoon. So here's the pericardial cavity, which we're going to talk a lot about. But that's a transcellular compartment. Here is a blood vessel. So some more fluid compartments. The intercellular fluid would be the fluid inside this cell, and this fluid inside the red blood cell. The blood plasma compartment, which is a member of the extracellular fluid compartment, lives in this blood vessel itself. And this, of course, is the interstitial fluid compartment. All right. The movement of body fluid. Water moves pretty easily, right? We know that already. Capillaries, it moves super easily through there, especially through the more leaky type of capillaries, the discontinuous and fenestrated capillaries. We've talked about those before. The continuous capillaries, a little stingier. Sometimes we need the aquaporins channels to move through these types of capillaries. What are the mechanisms? Well, they can slide. Well, they can use aquaporins channels. There's water channels. But they can slide between the cells of capillaries. They can go right through the plasma membrane of some capillaries. Remember the fenestrae stated capillaries, which the little holes got punched all over through in the capillary. They can go through there. So, all right, hey, that's it. All right, so let me shut down the shop here. I'll come back and field any questions. As always, email me your questions. I love questions. I don't get enough questions. And we'll see you later.